In this video, we are going to see about tumor markers and its types. And in types, CEA, PSA, and alpha fetoproteins. These are types of tumor marker we are going to see in this PPT. And we'll go to PPT. Okay, tumor marker. What is tumor marker? It is a chemical indication of presence of cancer called tumor marker. Okay, biochemical substance which present in a blood that indicates some disease. If any disease is there, some particles are elevated in a blood, like example, if patient have a diabetic, glucose level will be excreted, like way the tumor marker also elevated in a tumor condition. So biochemical indication, some tumor markers is elevated, will confirm that is any tumor in the body, and presence of cancer is called tumor marker. Abnormally produced by tumor cells such as surface antigens, cytoplasmic proteins, enzymes and hormones. The tumor marker might be anything from below these characters, such as surface antigen, cytoplasm, proteins, enzymes and hormones. Can be measured in serum. It can be measured by serum only, and useful for monitoring therapy response. If patient having a cancer, they are undergoing the treatment like chemotherapy and drugs, medications they are taking, that time we are monitoring the tumor marker for therapy response, how the medications work on as cancer types, and how will be controlling, that will be calculated by the tumor marker by calculating the therapy response. Types of tumor marker. There are two types of tumor marker is there. One is tumor specific antigen and another one tumor associated antigen. We are going to elaborate in this two topics. First, tumor specific antigens. It might be radiations. It may be caused by the radiations, chemical, carcinogens or unknown risk factors. Causes abnormalities of genetic information available in the cancer cells. It causes abnormalities in the genetic, which is a DNA damages, it will be created available in the cancer cells, and then synthesis new antigen specific to cancer cells. It will be formed new antigen. In the words, it means neo means infants, like children's, and antigens, we are knowing that foreign substance. It will be formed in neonatals or infants. That is the tumor specific antigen. And tumor associated antigen, it is also called oncofetal proteins. In a word, that means oncofetal means fetal proteins. It will be formed in fetal life, exist in both embryos and fetal tissues and cancer cells. Also cancer cells are present in embryo and fetal tissues. They are produced in large quantities in fetal life and released in fetal circulation. It will be produced large quantities in fetal life. Fetal life means when baby is in mother's placentals, there is high productions of a tumor marker, this types of tumor associated antigens, and it will be released in fetal circulations. After birth, it can be oncofetal antigens. It will be present in trace amount only in the adult stage. Okay, the onset of malignancy in adult life, synthesis of oncofetal antigens, fetal life, which are suppressed in adult life. Whichever tumor marker produced in a fetal life, it can be reduced, and it will be trace amount in the adult life. It again reactivated with malignant transformations of cell. It can be reactivated again and reappears of the circulations. It might be occur. Okay, the examples of oncofetal antigens, CEA and AFP. The full form of CEA, carcinoembryogenic antigen, and AFP, alpha fetoprotein. Okay, analytical criteria. The tumor marker has high specificity. Specificity in the sense that which tumor is came, that specific tumor marker also elevated. That what is meaning of specificity. And accuracy, precisions, same meaning. It will be accurately elevated for particular tumor. Okay, what tumor marker will be elevated in particular tumor, that will be accurately and precisionly calculated. Method should simple and easy to measure. Whichever method we used to calculate the tumor marker, it should be simple and easy to measure, and very low costly it can available in low cost. And clinical criteria of tumor markers should be disease sensitive. It must be positive in all patients with a particular cancer. Tumor marker will be positive. Then no false negative results should be provided, and should show increased levels of presence of micrometastasis should not show. Okay, next we are going to see about classifications of tumor marker. First one, tumor associated antigens. We are going early. This only we are seeing in early PPT. Carcinoembryonic antigens and alpha fetoproteins. Other antigens like tissue polypeptide antigens, pancreatic oncofetal antigens, colon specific antigens, beta oncofetal antigens. First one is tumor associated antigens, and second one is carbohydrate antigens detected by monoclonal antibody. More organ and tumor specific. CA means carbohydrate 125, CA 15 3, CA 99, CA 72 4, CA 50 and others. Mammary serum antigen MSA, MAM6, mucin like carcinoma associated antigens MCA. These are all carbohydrate antigens. And pregnancy associated antigens beta CG, beta SCG, human gonadotrophic hormones, chronic gonadotrophic hormone, placental like alkaline phosphatase, isoenzymes, and other antigens pregnancy specific glycoprotein, human placental lactogen, A2 pregnancy associated globulins, sex hormones binding globulin, steroid binding beta globulin. These are all pregnancy associated antigens. Next, hormone used as a tumor marker: PTH, calcitonin, growth hormone, ACTH, and other hormones such as insulin, glucagon, catecholamine, serotonin. These are all hormones used as hormones tumor markers. And enzymes, isoenzymes used as a tumor marker. Enzyme synthesized by the tumor marker: prostate, acid phosphatase, and prostate specific antigens, neuron specific enolase, LH isosomes, CK, LDH is a dehydrogenase, lactate dehydrogenase enzyme, and CK is a creatine kinase enzymes, glycosyl transferase 2 isoenzymes, enzymes derived from the organ tissue with metastatic carcinoma. From bone isoenzymes of ALP, osteoblast stage, ALP will be elevated in a bone. From liver isoenzymes of ALP from liver cells. Gamma glutamyl transferase GGT, 5-nucleotidase. These enzymes are elevated in a liver when the liver cells have the tumor. And from prostate, acid phosphatase ACP will be elevated. And miscellaneous of tumor markers: salicylic acids concentration in serums, polyamines, monoclonal immunoglobulins, steroid receptors like estrogen receptors, progesterone receptors, cellular markers, T lymphocytes, B lymphocytes. If you guys like my video, please subscribe my channel and hit the like button and comment. Thank you.